Hello students, we are going to continue with the next session, that is which is connected with a 3D printing technology of various organs. The organs which are synthesized are called as organoids. For that, in order to synthesize the organs by 3D bioprinting technology, we require the building blocks, what we call it as a scaffold, over which only our bioink is placed. So the overall development of uh, biology and engineering is applied. We call it as tissue engineering is a part of the field which is associated with your printing technology. Why we are doing this means due to lack of donors for the important organs like liver, heart as well as kidney. We don't have any donors for this, even if it is transplanted from allogenic transplant, that is other person, non-relatives, that is in turn imposes some rejection reactions, so that it is a success of the organ, transplanted organ rate is declining. So in order to overcome the difficulty, we are trying to uh, have a solution by tissue engineering, which is responsible for the production of our bioink as well as scaffold. So the tissue engineering is a field that applies the principle of biology and engineering for the development of functional substitute for the damaged tissues which is present in the various organs. So initially the cells are isolated from the patients and the cells are then cultivated in the cell bank. The different biomaterials are utilized to fabricate the scaffold. The next step is to seed the cells over the scaffold. Then the seeded scaffolds are then added with our growth factors. It may be hormones or cytokines. Then altered one is in turn, this is providing a real life environment for the growth and development of the tissue, so that we are creating a bioactive environment for this. Finally, the engineered scaffolds are then placed in the bioreactor, a device which mimics the condition inside the body. Then later, whenever there is a need, that is converted or utilized for the development of the organoid. So what are stem cells? Before going to the details, we must know what are all stem cells. The cells that can renew by themselves or self-renewal process. It can develop by on its own. They have the potential to differentiate into one or two different form of the cells or lineages, we call them as. The type of progeny stem cells are present. They are classified by Bernard and Paulson during the year 2004. So we classify the stem cells into three different types: totipotent, pluripotent and unipotent. Totipotent cells can produce all cell types, and pluripotent cells can produce most of the cell types, and unipotent can produce only one stem cells. For example, we come across the embryonic stem cells are called totipotent stem cells that can be differentiated into any cell that is derived from the fetus embryo. That is, aborted fetus tissues are taken for this purpose. And pluripotent stem cells, example we come across hematopoietic stem cells are called as pluripotent stem cells that can differentiate into WBC and RBC. And unipotent cells, if it is a neuron, we can that ectoderm can be converted to neuron, and mesoderm can be converted to blood cell, and endoderm can be converted to liver cells. But if it is an hepatocyte, we can produce only hepatocytes only. If it is a neuron, we can develop only neuron, like that. Or if it is an cardiomyocytes, we can develop only cardiomyocytes. That is unipotent in nature. What do stem cells they are looking like? Normally they are spherical cells about 6 to 8 micrometers in diameter, and they don't have any morphological features. They will not have a perfect shape. They will not have the shape like a neuron or nephron or whatever it is, like any other cells or cardiomyocytes. But they divide rarely, and they commit to differentiate in a stochastic manner. That rate is 1 per 6 in week. 1 per 6 weeks. The rate of development is very slower in nature. What are all the tissues we have? The stem cells. In our body, we have rapidly proliferating tissues such as bone marrow, small intestinal epithelium, muscle, and skin. They are appear to have stem cells. The same seems to be organs true for with the slow turnover times, such as liver, brain, and even beta islets of the pancreas. The stem cells that build tissues. The stem cells are committed to differentiate into undergoes different organ functions, repair and formation. So normally organ development process, a repair process, a repairing mechanism where we make use of the stem cells for this purpose. The cellular fate process of this cell proliferation, cell death, cell movement and cell differentiation together to form the fate of the committed cells. The embryonic stem cells. First and foremost, embryonic stem cells are derived from the human peri-implantation embryos formed during the infertility treatment of the couple for in vitro fertilization. The unsuccessful tissues are taken for this purpose. These embryonic stem cells can self-renew apparently without limit in culture and are pluripotent. They can give rise to any type of cells in the body. The central issue that is associated with your tissue engineering products derived from your embryonic stem cells is immune rejection and based on mismatch of the genetic histocompatibility loci which is present on it. Therapeutic cloning offers a potential means to generate cells with exact genetic constitution of the individual patient. That is normally what we do is, in order to avoid the rejection reaction, we take up the autologous cells, that is autogenic cells. Autologous cells mean the cells derived from the same patient itself and is differentiated and is injected to the patient for the treatment procedure. The clinical application of embryonic stem cells for tissue engineering depends on the development of robust method to isolate and grow them under conditions consistent with good manufacturing practice and regulatory reviews for the safety issues. The success remains in the case of the embryonic stem cells. We should differentiate them into desired lineage with high efficiency. Adult stem cells are undifferentiated cells among the differentiated cells in the tissues and organ that can renew and yield the same some or major type of the cell types which is present in the tissue or organ. For example, we come across the bone marrow contains our hematopoietic stem cells that can differentiate into RBC and WBC. They are called as adult stem cells. Induced pluripotent stem cells, we call it as iPSC. Induced pluripotent stem cells that are isolated from the adults that are genetically reprogrammed like embryonic stem cells, and they are forced to express the gene and factors important for the maintaining the properties of the embryonic stem cells. So what we do is we convert or we push back our adult stem cells back to the embryonic stage by adding some growth factors and other details which is necessary for a reprogramming process. So such pluripotent stem cells are called as iPSCs. That is induced pluripotent stem cells. That is of clinical significance. This was developed in the mouse during the year 2006 and in the human later they reprogrammed human cells also during the year 2007. More additional research is needed in the case of iPSC, induced pluripotent stem cells, because they are useful in drug development and modeling of the disease. But problem lies here is that this reprogramming process only to repair the damaged tissue in the human body. On reprogramming process, it should not transform itself into a cancerous or a tumorous cell on treatment process. There are major differences between embryonic and adult stem cells that already we discussed. They are limited in the case of the adult stem cells, but embryonic stem cells are more advantageous when compared to the adult stem cells. That can be embryonic stem cells are easily grown in the culture and compared to adult stem cells, and moreover it is difficult to isolate the adult tissues. That is a challenging job, and to culture them also. Cellular differentiation. What is cellular differentiation means? The process in which the cell changes from one type of cell into the other type of cells. For that it requires various growth factors. So the growth factors includes our various cytokines as well as hormone for the purpose of this. Here is a diagram which shows how the myeloid differentiation that takes place in the presence of various growth factors into DC, LC, macrophage, osteoclast, neutrophil and eosinophil. What are all the cell sources? Already we discussed autologous, autogenic, allogenic and xenogenic. Autogenic comes from the same persons to form the new cells. Allogenic comes from the body of the same species, that is kith and kin. We call it as allogenic or relative, or it may be non-relative in nature. And xenogenic, it comes from the different species. It may be from any animal source for that matter. Where to grow these pluripotent or induced pluripotent stem cells for the growth and development? In the case of tissue engineering means we require in order to provide the perfect shape to the organ, we require a three-dimensional material to provide the structure for the particular organ, and where the cells will go and adhere themselves and proliferate and secrete extracellular matrix. So such three-dimensional materials are called as scaffolds. There are different types of materials have been used to construct scaffold, such as polymers such as PLA, PGA, PCl and PEG. And further, bioactive ceramics like HA, TCP also used along with our natural polymers like collagen, GAGs and chitosan. The scaffold the construction requirements. In order to have a perfect shape, we have to create the three-dimensional structure of the organ by scanning MRI CT scan, as well as in the case of the 3CAD, that is three-dimensional computer aided design is utilized for the purpose of this. Then the particular shape of the scaffold is constructed by using our biomaterial. And moreover, this biomaterial or scaffold should provide support for the endogenous cells to attract and grow and differentiate in the in vitro culture, as well as after implantation also it should provide the support for it, so that the biomaterial used to fabricate the scaffold and provides the necessary environment as if it is in the natural environment in the particular organ. The scaffold should provide bioactivity also. The scaffold interacts with the engineered tissues and facilitates regulates the activity of the particular organ. It creates a cell adhesive ligands so that it can attach itself and to create the morphology of the particular organ. It also acts as a delivery vehicle, reservoir for the exogenous growth stimulating signals such as growth hormones and cytokines. It provides the mechanical property for it. The intrinsic mechanical property of the biomaterials are given by this scaffold for the development of the cells. Normally, epithelial cells, fibroblast, muscle cells, neurons, they sense the stiffness on the substrate and show dissimilar in the morphology. So it should be more biocompatible when compared to the normal plastic like devices, that is prosthetic devices. A scaffold synthesis, number of different methods are introduced for the preparation of the porous structure in the scaffold by the tissue engineering. Each of these technique has own advantage. A nanofiber self-assembly is done, which forms the extracellular matrix. It's a crucial step for the development. Moreover, the hydrogel of the scaffolds have been superior in the in vivo toxicological studies are done so that the macro scaffolds are animal derived in nature. Textile technology. This technique includes all the approaches successfully employed for the preparation of non-woven meshes of different polymers. But it has the drawback, it has difficulties in obtaining high porosity and regular pore size. The scaffold is synthesized by freeze drying technique, electrospinning. Freeze drying technique is the polymer is dissolved in the suitable solvent and later it is mixed with water and the polymeric material is obtained as a element emulsion, then it is added to the mold quickly and it is frozen and it is immersed into the liquid nitrogen. Frozen emulsion is subsequently dried to remove the dispersed water and solvent, leaving the solidified as the porous polymeric structure. Electrospinning is the method whereby a fiber structure is made, the fiber mesh is submicrometer or nanometer level, that is using the electrospinning method, just like a thread. It will create and it is spun over the particular shape and we are creating the fibrous material, a thin fibrous stream. This fibrous material is molded into perfect shape with the help of voltage. A power supply of 30 kilo voltage is used. Using a syringe, it is from the tip of the needle, it will be injected and it is converted to the particular shape of the scaffold for this process. So, various biomaterials are utilized for the scaffold synthesis, natural biomaterials and synthetic biomaterials. The natural biomaterials includes protein origin biomaterials and polysaccharide origin biomaterials. The protein origin biomaterials include silk, collagen, fibrin and gelatin, whereas polysaccharide origin biomaterial includes hyaluronic acid, alginate, agarose and chitosan. The polymer biomaterials and ceramic biomaterials. The polymer biomaterials includes polyethylene glycol (PEG), polyglycolic acid (PGA), polylactic acid co-glycolic acid (PLGA), and polydilactide (PDLLA), and polycaprolate caprolactone, that is PCl. Sorry. And ceramic materials include alumina, zirconia, sintered HA, tricalcium alpha beta tricalcium phosphate, and tetracalcium phosphate, hydroxyapatite, bioactive glass, calcium phosphate, etc. Bioreactors. The tissue engineering bioreactors are defined as the devices that uses mechanical means to influence the biological process. The bioreactors are nothing but in which the cells are grown and it is either converted to hydrogel for the formation of the bioink or extrusion material we discussed. So bioreactors can be useful in vitro development of new tissue by providing biochemical and physical regulatory signals for the cells to undergo differentiation and to produce your extracellular matrix before to the in vivo implantation of the particular organ. So the bioreactors are mechanical devices, mechanically stimulates the cell and it encourages the cells to produce extracellular matrix in the shortest period of time and moreover it is homogeneous in nature. The PGA scaffold is seeded into these articular chondrocytes that expresses significantly improved ACM proteins in the cultured condition under hydrostatic pressure. This is a successful culture method in the shortest possible time where we can able to achieve it. Various type of bioreactors are available for the production of the culture and the use of the cells. So it provides cellular spatial distribution. The scaffold sizes ranges and can be easily fabricated during this process in the bioreactors. According to Munjak and Novakic, bioreactors are designed to perform at least one of the following function. What are all the functions? Let us see. It provide a spatially uniform cell distribution. It maintains the desired concentration of the gases and nutrients for the growth and development of the particular cell, and it is responsible for the mass transport of the tissue, and it is useful for the construction of physical stimulus, and it also provides information about the formation of the 3D structure of the tissue. Here, here is a bioreactor design requirements. There are various bioreactor design how to behave. It should be biocompatible, bioinert in nature. Normally, it is made up of stainless steel and titanium, so that it should not interact with the seeded cell at any cost. It should not invite any infection of the microorganism on the development process. It should maintain the 37 degrees centigrade and humid atmosphere as if the cells remain in the normal body condition, that can be sterilized and reused. The materials with the different properties are needed for various components in the bioreactor. For example, transparent materials can be of benefit in allowing the construction and monitoring of bioreactors. So when the bioreactor is transparent, only when we can clearly view whether the cells are being constructed over the scaffold or not, that we can identify very easily. It should provide proper parameters of the environment such as pH, temperature, nutrient concentration, oxygen level that can be monitored, and the sensors should be incorporated into the design, and it should also for requires forces such as cellular stimulation that are very small enough to and it is important to ensure the pump motor should have the capacity to apply this into using the smaller forces. There are numerous types of bioreactors are available that are identified by the tissue engineering applications, that includes spinner flask bioreactor, rotating wall bioreactor, compression bioreactor, strain bioreactor, and hydrostatic pressure bioreactor, and flow perfusion bioreactor. A spinner flask bioreactor induces mixing up of oxygen and nutrients thoroughly throughout the medium and reduces the concentration of boundary layer, so that all the cells which are present in the medium acquires oxygen as well as the nutrients. The scaffolds are suspended at the end of the needle of the flask of the culture media. The magnetic stirrer that completely rotates so that it allows the movement of the fluid which is present in it. Normally, the size of the spinner flask is around 120 ml in volume and runs at 50 to 80 rpm, and 50% of the medium is changed every 2 days. Here we can see the video. Here we can see the spinner flask bioreactor where a stirrer is found inside that is rotated, and we can see the inlet and outlet of oxygen and nutrient supply, where after 2 days the nutrients are eliminated in a magnetic stirrer is available, and we can see the cells are gets mixed up by this process. Rotating wall bioreactor, it is developed by NASA. The scaffolds are free enough to move in the media and the vessels. As the rotating wall vessel bioreactors, it is cylindrical chamber. It rotates like a drum, rotating drum. So the inner and outer wall are capable of rotating at constant regular speed. So the vessel is then rotated at a regular speed so that the balance is reached by the gravitational force during this process. So that the scaffold which is remaining in the medium gets suspended, that allows the entry of the cells, and it is during the rotation process the exchange of nutrient. It allows the exchange of nutrient as well as oxygen to the tissue properly. On stoppage of the rotation temporarily, by adding a fluid pump, the media is constantly pumped throughout the vessel. The gas exchanges also made possible in this bioreactor at a speed of 15 to 30 rpm. Here is the diagram which shows the rotating wall bioreactor. Compression bioreactor. This class of bioreactors are generally used in cartilage engineering for this purpose. This is compression bioreactor consisting of the motor. It's a linear movements that controls a mechanism. A signal generator can be used to control the system, including load cell, whereby transformers are used to measure the load response and imposed displacement. Here we can see that this is a compression bioreactor, the head for dispensing the pressure and scaffold constructs which is available. Strain bioreactors. The strain bioreactors are used for different types of tissues such as tendon, ligament, bone, cartilage and cardiovascular tissues. These designs are very similar to the compression bioreactor. Only difference is that the force is transferred to construct. Instead of platens as in compression bioreactors, a way of clamping scaffold device is needed for this process in this case. Here we can see the clamping process, how it is involved. Uniaxial strain bioreactor for the engineered ligamentations. Hydrostatic pressure reactors. They are useful in cartilage engineering. It can withstand very high pressure when it is applied by means of any mechanism. The variation in the pressure is water filled pressure chambers are available. That pressure is a media filled in the chamber via an impermeable film so that it moves backward and forward. Here is a diagram for it. This is a hydrostatic pressure reactor. Flow perfusion bioreactor. The flow perfusion bioreactor is shown to provide more homogeneous cell distribution throughout the scaffold. So in comparison between flow perfusion and spinner flask and rotating wall bioreactors, flow perfusion bioreactors are the best for the fluid transports, whereby the media containing growth factors are unable to move here and there and can reach the interior side of the scaffold for the growth and development of the cells in this case. The media can be replaced often by the media reservoir. Here we can see the diagram of it. And moreover it is transparent, where we can see the development of the scaffold also. Here is a perfusion bioreactor where we can clearly see the development of the scaffold containing tissues. What is bioactive environment? Bioactive environment is creating an environment similar to that which is present within the organ inside our body. So we are creating an artificial environment, artificially the environment which is present in our body for the growth and development of the particular uh, stem cells. It may be natural stem cells or it may be reprogrammed stem cells or iPSC. So what are all the things we require for it means for the bioactive environment we require growth factors, hormones as well as cytokine for this development. Here is the pathway where the cells undergo development which involves ERK pathway and JAK-STAT pathway for this purpose. So the growth factors are the proteins that are provided by the body normally that regulates the cell division and cell survival. So these are developed by our genetic engineering lab and used for the biological therapy. The growth factor binds to the receptor on the cell surface and activates the cellular proliferation and differentiation. They are quite versatile and differentiating into numerous different cell types. Various growth factors are secreted by the body that are inoculated into the medium of the cell culture that includes EGF, epidermal growth factor, FGF, NGF, PGDF, platelet derived growth factor, IGF, insulin like growth factor, BMP, HGF, GDF, and many others. So hematopoietic stem cells growth factors are hormone-like substances that stimulates the bone marrow to produce the blood cells. Shortage of the blood cells are causes most of the symptoms in people with MDS. Use of growth factors is very appealing. Erythropoietin is a growth factor which is necessary for the development of RBC. Interleukin 11 that stimulates the platelet production after chemotherapy. The sequence of amino acids permits the growth factors to be placed in the family. So normally the insulin family comprises of somatomedins A and C, insulin-like growth factor IGF, and multiplication stimulation factor MSF, and a second family consisting of sarcoma growth factor, transforming growth factor SGF, TGF, and epidermal growth factor. Here we can see how it is associated, how the growth factor binds on the cell containing surface receptor and it activates the cascade of protein kinases that enters into the nucleus where it activates our RB retinoblastoma E2F elongation 2 factors E2F and RB is phosphorylated, then E2F is activated that in turn activates our various CDK cell dependent CDK cell cyclins and further that is associated with our cell division process. Hormones. Hormones are the chemical messengers that travels into the blood stream that reaches the particular glands and tissues and organs that maintains the homeostasis for the development of the cell. Hormones regulates the function of the particular target cells and expresses the receptor for the particular hormone. Normally the cells have the receptor for the binding of the hormone. So normally for a particular organ the target tissue will not be associated with the same endocrine organ itself. It is far away tissues are benefited by this. For the movement from the endocrine gland to the target tissue, it requires the transport medium is nothing but our bloodstream which carries along with the plasma protein it reaches. We know the role of insulin as well as glucagon. So various two major classes of hormones are available. One is proteinaceous hormone and steroidal hormones. Already you studied under here endocrinology. These hormones are able to work on the metabolism, it acts upon the metabolism for the development of cells. Here we can see that mechanism of hormone action. Cytokines. Cytokines are small secreted proteins that are released by the particular cell, which has effect on the interaction and communication between the cells. How the cells are communicating with each other means the just like your message, WhatsApp message மாறின்னு வச்சுக்கோங்களேன். So these cytokines are general names. They are include lymphokines, monokines. Lymphokines are made from lymphocytes and monokines are made from the monocytes. Chemokines are chemotactic activities, and interleukins further they are derived from our leukocytes. The cytokines are of a many cell population in nature. The predominant producer of the cytokines are T helper cells and macrophages. We have various pro-inflammatory cytokines are produced by the macrophages that are involved in the upregulation of the inflammatory reactions. They are abundant. There is an abundant evidence that pro-inflammatory cytokines such as IL-1 beta, IL-6, TNL, TNF alpha are associated in the process of pathological pain. The primary cytokine that is released by the monocyte and macrophages are nothing but here IL-1 beta. It is also by the non-immune cells such as fibroblast, endothelial cells during the cell injury, infection, invasion and inflammation. IL-6 is shown to play in the central neuronal reaction in the nerve injury. The suppression of IL-6R by the in vivo population, anti-IL-6R antibodies are used to reduce the regenerative effects. Normally, the IL-6 are involved in the activation of microglial and astrocyte cells for the regulation of the neuropeptide expression for this process. These anti-inflammatory cytokines are a series of immunoregulatory molecules that are responsible for pro-inflammatory cytokine response. They are associated with our human immune response in this case. So the major anti-inflammatory cytokines includes IL-1, IL-4, IL-10, IL-11 and IL-13. Further, we have TGF-alpha, that is transforming growth factor beta, are also acting upon leukemia inhibitory factors. Here is the diagram which shows the various cytokines that are associated with our cell proliferation process. What is the application? The application of this tissue engineering is involved in the production of their 3-dimensional printing of the organs and breast reconstruction and cardiovascular system, cardiac tissue engineering, blood vessel production, heart valve production, musculoskeletal system, bone generation, bone and cartilage reconstruction, production of nervous system and skin. Here the breast for the people with surgery after mastectomy or lumpectomy, that is any lump or cancer is available in the breast region that are in turn removed, then later the soft tissues are in turn replaced with our breast reconstruction process, in which the pre-adipocytes, fibroblast, smooth muscle cells, muscle myocytes and chondrocytes are utilized for this purpose. The 3D polymeric materials provides the support over which the cells are added, and it is surgically implanted to the delivery of the cells are done by the hydrogel material or bioinjection during this procedure, so that the breast is reconstructed which looks like a normal one, which doesn't have any evidence of any surgical removal of the breast by the cancer process. According to ACS, each year 182,000 new cases of breast cancers are detected that are potentially required breast reconstruction process in the US. The US breast cancer treatment of United States, they spend 8.1 billion per year for this process. So there are two ways by which the breast is reconstructed for this process: in situ adipogenesis and de novo adipogenesis. In situ means the first technique that employs the transplantation of the supporting matrix over which the growth factors and other things are added. The fibroblast and pre-adipocytes are added to form the matrix. Then the extracellular material is developed over the scaffold, and later it forms the perfect shape in the case of this. For the perfect shape of the breast reconstruction procedure, it should be elastic and tensile. For this purpose, we are using the tissue engineering method in all possible ways, involving fibroblast, smooth muscle, skeletal muscle, and elastic cartilages. And de novo adipogenesis, we place the implants and we inject our Bioink containing all these tissues, then we allow it to grow. We don't know how it will grow. We never add our bio environment containing growth factors like hormones, cytokines, etc. As such, the body provides the necessary material for the process. The implantable form of the materials what we use, the supporting media or scaffold is involving foam, sponge and films, and solid devices are utilized for this purpose. The cells are injected in the form of gel, hydrogels and as well as microbeads. The natural polymers such as alginate, chitosan, hyaluronic acid, collagen are used as the injectable material. By using the syringe, it is implanted for this process. This is a cardiac tissue. The heart tissue remains leading heart disease, the leading cause of the death in the developing countries as well as developed countries. Along with stroke, the principal CVD accounts for the 40% of the all death. In order to replace the damaged tissue in the myocardial region, we make use of this direct implantation technique where we inject the cells as well as the scaffold for the regenerative process. The myocardium is a highly differentiated tissue. It is about 1.3 to 1.6 centimeter in thickness in human, composed of cardiomyocytes and fibroblast. It supports the formation of the vasculature, that is development of blood vessels and collagen based extracellular matrix. So only 20 to 40% of the cells in the heart are cardiac myocytes. It occupies 80 to 90% of the heart volume. So the average cell density that is created in the rat model includes 5 into 10 to the power 8 cells are added for this purpose. There are some problems associated with this in the developmental process. Since it mimics the native myocardial, but the stability of the cells we have to take into consideration, since it is still it is trial and error. In getting the adult cardiomyocytes, and it is difficult to proliferate and utilize for the autologous engineering process, that it will take longer time for treatment procedure. So we have to take into consideration in the case of the critically ill cardiac patients during this procedure. or we can make use of only either cardiac patch or the cells from the other sources, not from the autologous. It may be allogenic or xenogenic cells can be utilized for this procedure for the treatment of or implantation of the cardiac tissue. Cardiac tissue engineering cell sheets are prepared. Shimizu during the year 2002 he created neonatal rat cardiomyocytes and implanted a temperature sensitive ply-in isopropylene acrylamide surface. The change in temperature from 37 to 32 degrees centigrade causes monolayer to detach. The initial surface area decreases during the detachment procedure. And further the cell sheet increases approximately 20 micrometer to 40 micrometer during this procedure. When the cell sheets are laid over each other, the electrical communication was established so that the cell starts beating spontaneously. So when it was implanted successfully into the nude rats, the shell sheet survived for about 12 weeks and caused vascularization. Here we can see the development of your cell tissue that can be added as a patch for the transplantation process to replace the damaged cardiomyocytes. A biomimetic approach where we are trying to create biomaterial and bioreactor that can able to contract and mimic like our cardiac tissues. That is in progress. And we are also producing a blood vessels by the tissue engineering process that are utilized in vitro as well as synthesized in vivo as well as in vitro. Alex Carroll, the father of vascular surgery, he has tried this method. uh, Cardiovascular has become more prevalent in 100 years. So in the cardiac patients, we go for transplantation of the autogenous veins and arteries that are provided by the patient itself. The suitable autogenous vessels are taken for this purpose and it is utilized. It was successful that prevented many persons from early death. What we do is here we take up the vascular grafts and we improve the vascular graft by placing in the patient's variable like diabetes, renal failure, all the cases for proximal and distal angioplasty is done to increase the lifetime of the person. In order to create the blood vessels, we are making use of polyethylene terephthalate as well as expanded polytetra fluoroethylene that are utilized for the prosthetic vascular graft to make the people survive during the angioplasty. Blood vessels, the tissue engineered blood vessels are shown here. How we are creating it. In vitro tissue grafts are developed by TEVG in vitro method, what we are seeing in the diagram. Here we can see the tissue engineering vascular graft. Heart valves. Heart valves is a considerable medical problem around the world. 87,000 heart valves replacement is done so far by the American Heart Association. It is used to, in the end stage of heart valve disease, it is mandatory to replace the valve because it fails to contract and relax. So it loses its flexibility property. So in order to overcome this, we are making use of tissue biomechanics, immunology, immune response, cellular and tissue development, chemical, physical, and pharmacological manipulation of this heart wall development. Here we can see the advantages of the heart wall development by tissue engineering. In order to prepare the heart walls, we are creating a biocompatible and biodegradable highly porous material in which the scaffolds are made by the natural and synthetic material made up of collagen, fibrin, elastin, glycosaminoglycans, and decellularized heart walls, where it provides the necessary environment, that is extracellular material components for it. Further, the synthetic material make use of polyglycolic acid, PGA, polylactic acid, polylactoglycolic acid, and polyanhydrides and polypeptides for this purpose. And polyhydroxy alkoinate is also utilized for this purpose. Here we can see the development of the heart wall by the 3D bioprinting. So normally the scaffold is needed for the controlled growth and development of your heart walls, which can open and close like a tricuspid and bicuspid wall, which is present in the natural environment of the heart for the pumping function of the blood. Here we can see the successful development of tissue engineered heart walls in end stage wall disease. And bone regenerations, a scaffold when bone is defective or bone damage is done. Scaffold based tissue engineering is capable of treating. Optimized cells are attached to the maintain bone marrow cells. Allogenic culture are expanded and are utilized for the treatment procedure. Here we can see the overall picture. How the bone marrow cells are injected to the patients by the invasive procedure. It is more advantageous. Bone and cartilage reconstruction can be done in vivo as well as ex vivo gene therapy is done for the patients. Here bone and cartilage reconstruction. Here is the producing 3D structure of the human ear shape by the genetically engineered nude mice. This is the animal model, successfully created model of the ear, extra ear. For the construction of the cartilage, we require PGA, polyethylene, polypropylene hydrogel and chondrocytes are utilized for this purpose. And brain implants are also made by our bioprinting technology. This is utilized to treat the patients of Parkinson's disease, Huntington's disease, and epilepsy and stroke. Here is a wired therapy that is utilized for this purpose to calm down the symptoms of Parkinson's disease in patients of the disabled overactive nerve cells. And combined replacement regeneration implants are also developed to treat other disorders. Skin and wound repair. Normally, any damage on accidental cases, the skin is injured. In a larger cases, that may invite infection in some of the cases. In severe case of diabetes mellitus, the skin is injured due to infection. That cases can be chronic. 7 million wound cases are generated. They are chronic in nature. Such people are treated with the skin wound repair mechanism. Here we can see the new approach where we make use of our hyaluronan and non-sulfated glycosaminoglycans are utilized for tissues, human tissue and wound repair mechanism. Here we can see that the diagram which shows how the scaffold can able to treat the skin tissue. Here is the tissue where we can see the second skin is developed that can be applied over the wound for the wound repair mechanism. That's all.